maamuzi ya kibinafsi. Kwa hiyo siwezi kulazimisha mtu anione mzuri au siwezi
Yes. Good evening, everyone. Welcome to this CME. Today, I'm Frank Okada Nyakawa. I'm an ophthalmologist from Nyamira County. I'll be waiting to present, to introduce the moderators. So we are kindly waiting. There's a hitch up. We are waiting for Dr. Kibata to be ready, and then we start the CME. So kindly be patient. Thank you.
Good afternoon, everyone.
Good afternoon.
Okay. Good afternoon, Dr. Kibata.
Good afternoon, Frank. Sorry I'm late.
Yes, let me introduce you. Okay. So welcome to this CME today. Our moderator will be Dr. Kibata. We will talk about why we need to take the AstraZeneca vaccine. So Dr. Kibata is an ophthalmologist. He's also a VR surgeon. He's also the medical director of CTI Hospital. So he'll moderate and he'll introduce the speaker of the day. I'll come back again to the questions, the QA time. Thank you. Welcome, Dr. Kibata.
Thank you, Frank, and welcome everybody. Just a clarification, I am not here to say we should take the vaccine, but I think I'm here to lead the discussion, hopefully to answer many questions that members have. And with us this evening is Dr. Vincent Muturi Kioi. He's here courtesy of Dr. Mushai, and we want to say a special thank you to Dr. Mushai. I think from what I heard, they have been classmates for a long time. Vincent is a medical doctor working with the clinical development department of International Vaccine Initiative. He's responsible for protocol design and safety, monitoring of clinical trials, evaluating biomedical prevention interventions against HIV and other diseases of public health importance. He also assists in the design and implementation of epidemiological studies aimed at providing data to be used for the design of clinical trials and participate in the clinical aspects of the training and capacity building programs in the AVI African Clinical Research Center Network. He has 12 years of experience in vaccine clinical development and life cycle management. He has worked at GSK, he has worked in Cambridge, CDC. He has worked basically in all the important places. He received his MB CHB from the University of Nairobi, DTMNH from the London School of Hygiene and Tropical Medicine. He has a Master's of Science in Vaccinology. I didn't even know that exists. And a pharmaceutical clinical development from the University of Siena in Italy. Basically, if there's someone to tell us about vaccines, this is the man. So, Daktari Muturi, please, karibu sana. I think we are all eagerly waiting to hear from you, and the floor is yours.
Thank you very much, Dr. Kibata. Can you hear me? Can you hear me?
We can.
All right, thank you very much, and thank you for the opportunity to present today. So, as has already been said, my name is Vincent Kioi, and I work with an organization called IAVI. IAVI is a nonprofit organization which is focused on development of vaccines and other products for management and prevention of diseases that we consider to be of public health. My invitation from Mushai and the team was based on the fact that he thought that it would give an opportunity to for the team questions, or members of the association of the society to ask questions and seek clarification on different aspects of the vaccine, whether it be related to its safety or its efficacy. And so please feel free to ask on that. But I would like to say up front that I am not representing either the institutions or the organizations that developed the vaccine, nor am I representing the government that is responsible for the rollout of the vaccination program in Kenya. So all the responses I give will be in my own capacity as a healthcare provider and a health practitioner in Kenya, and to you as individuals who be seeking the information on your own behalf. So I'll just share my screen. I had prepared a few slides. These are mainly designed to just highlight the data that was used to support the use of the currently available COVID-19 vaccine that we are using in Kenya. As you may be aware, at this time in Kenya, we are using a single provided by the Serum Institute of India. The vaccine we are using is exactly the same as the vaccine produced by AstraZeneca that was developed by the University of Oxford. And so the safety and efficacy information that was used to license or to provide emergency use authorization for the Oxford vaccine is applicable to this vaccine as well. So the AstraZeneca team simply transferred the technology to the Serum Institute so that they could use their capacity to provide vaccines for countries around the world that would otherwise not have had access to these vaccines. Let me know when you can see my screen.
Yep, it's up.
All right. Okay. So I'll start with a disclosure. So as Dr. Kibata said, I'm a full-time employee of IAVI. Although I'm not employed by the University of Oxford, I'm currently working in collaboration with them on development of a HIV vaccine candidate. And this vaccine candidate actually uses the same chimpanzee adenovirus vector that is used in the COVID-19 vaccine, which I will discuss a bit as I go through my slides. And I have previously worked in the pharmaceutical industry for GSK vaccines, focused on pediatric vaccine development. This is the outline talk. I'll give a brief overview of coronaviruses, what we've known about them for some time, as well as talk about the burden of disease based on the currently available numbers and the case for the vaccines around the world to sort of interrupt or halt the pandemic. I'll give a brief overview of the process we go through to develop vaccines, which are aiming for licensure and widespread use around the world, and focus on the vaccines that have been developed against SARS-CoV-2, just a brief overview of the ones that are available in other parts of the world, in addition to the AstraZeneca one, and then focus most of my talk on the clinical development of COVID-19 vaccine from the AstraZeneca team. And I'll touch a little bit about the post-approval safety signals, which have become a contentious issue that has been in vaccination programs using this vaccine being halted in some parts of the world. So very briefly on coronaviruses in general, these are a large family of viruses that circulate in mammals and birds, and there are several types that have been known to infect humans since the discovery of viruses in the last century. So there are 7 types of coronaviruses that infect humans. These are responsible for the common cold and are present in causing a variable burden of disease from the common cold in different parts of the world. At the moment, for diseases caused by the coronavirus, the different coronaviruses that have existed in humans, there's no definitive treatment or vaccine that had been developed prior to this past year. Over the past years, we've seen novel coronaviruses shift from animal hosts to humans and cause outbreaks that have been largely localized and that have been controlled before they could result in the type of pandemic we are seeing now. So in the early part of the last decade, we saw reports of the Middle East respiratory syndrome, which was also caused by another beta coronavirus. This is associated with infection of individuals who had close contact with camels, which were the primary host of that virus. For that particular virus, the opportunity for human-to-human transmission was much less than it is for what we are experiencing now with the SARS-CoV-2. And although it had a very high case fatality rate, so nearly 35% of those who were identified as being infected ended up dying because it was not very well, it didn't have a very good ability to transmit from human to human. We noted less than 1,000 deaths worldwide from just over 2,494 cases as of 2019. Similar to what we are experiencing now, there was the severe acute respiratory syndrome virus, the first SARS, which came into being in China in 2002. This also had a much higher mortality rate than what we've experienced with SARS-CoV-2. So the mortality rate was above 9%, the case fatality rate was above 9.5% for the original SARS virus. But the rate of transmission between humans was not as high as what we've seen with the currently circulating coronavirus. So when looking at what's the case for the use of vaccines, although the case fatality rate we are experiencing with SARS-CoV-2 isn't as high as what has been experienced with previous, with other forms of coronavirus that have jumped to human novel infection. It's still quite very high compared to what we've seen with other respiratory tract infections that have this ability to spread at such a fast rate across the world. As of the figures being reported yesterday on the Worldometer, we have more than 121 million reported cases from all over the world and more than 2.6 million deaths. So this should tell you two things. Number one, the case fatality rate is much higher than you would expect with what it has been compared to by some people, which is the seasonal influenza virus. For the seasonal influenza virus, we typically see case fatality rates of 0.1 to 0.2. Here we are seeing case fatality rates between 1 and 2%. And these can be much higher in those who are most vulnerable to severe disease. At the moment, we've seen development of more than 48 vaccines which have gone into the clinic, and about 11 of these have reached advanced stages of development. That meaning that they're going towards the stage that would allow them to receive a license once the data is reviewed. 7 vaccines have so far been made available for public use. And as of earlier this week, on the 17th of March, reports were that about 400 doses had already been administered in 132 countries. This wide, large-scale vaccination program, which some are saying is the single largest vaccination program that's been implemented to address one entity at a single time point, is being put in place because the belief is that the figures to the left of your screen are only bound to get worse unless such a massive intervention is put in. This table summarizes some of the vaccines that have gone through advanced development and have become available for use by the general public around the world. On the third row, you could see some of the details on the AstraZeneca University of Oxford vaccine. So the vaccine type, it's a viral vector vaccine. The efficacy that was based on the data that was used for its authorization for use was about 70%. The number of doses that are recommended for that vaccine is 2 doses. And when looking at storage, which is an important factor to take into account when determining where it can actually be delivered, it has a storage requirement of about 2 to 8 degrees Celsius, which allows it to fit into almost every cold chain that's been in place around the world. When looking at the other vaccines that are available in other parts of the world, there are the mRNA vaccines that have been developed by companies like Pfizer, BioNTech, as well as Moderna. These have a much higher reported efficacy from the trials they've done, exceeding 90%. Both of these also require a 2-dose regimen. Unfortunately, the cold chain requirements for the mRNA vaccines is something that's out of the realm of possibility for most countries around the world, requiring storage temperatures of -20 to -70 degrees Celsius. Aside from what's been developed in Europe and the US, the vaccines from China and Russia, and these were very early on. So for example, the Russian vaccine, which is commonly known as Sputnik V, was the vaccine to be administered to humans. Sorry, I'm getting a lot of feedback. Could you go on? I'm not sure who that is. Thank you. The Russian and Chinese vaccines were used very early on while the vaccines were still in relatively early stages of development. And so they had been rolled out as the clinical trials were proceeding. And you might have heard that they were in use much earlier than what was available from other parts of the world. As I said earlier, we've seen vaccination programs being rolled out across the globe. Fortunately, we seem to be playing catch up when looking at this map, highlighting where vaccines are being used at the fastest rate, as well as where the uptake is highest. You see that although several countries in our region, including Kenya, have introduced vaccination programs, the uptake is still less than 1% of the population, which contrasts markedly with what you're seeing in North and South America, as well as in some parts of Asia and Russia. So when thinking about the development process that goes into preparing vaccine products to be used widespread on a large scale in multiple countries and at the community level, what you see is that typically what we have had in the past is a situation where clinical analysis in animals is carried out over sometimes months to years. And this follows a much more difficult process, which we call the discovery process. So that discovery process usually involves in the lab, usually people who are specialists in biology or of those particular pathogens, looking for what parts of the pathogen, in this case a virus, could be used to develop an effective immune response. For some viruses, such as the SARS-CoV-2, this has taken a very short time. So the initial sequence of the SARS-CoV-2 virus was provided by the Chinese in January last year. And by March, several laboratories around the world had already produced sequences for the surface proteins that they believed would be protective. The discovery process for effective vaccine targets can be longer. So I mentioned that I work in HIV vaccine development, and this process is still ongoing for HIV after 30 years because HIV is a much more difficult pathogen to develop vaccines against. Once we get into the preclinical phase, this is generally done to assess whether, number one, it's safe, and secondly, it can elicit an immune response that could be protective. And we typically see that going through several stages, from small like mice and rats to much larger mammals like non-human primates. And this could take several years before you get approval to move into humans. Once we are in humans, we typically start with very small phase 1 trials just to assess whether there are any unacceptable safety events. So we have vaccination of maybe 10 to 50 individuals and follow them up over a period of about 6 months to a year and look and see any problem that we can identify. At the same time, we see what kind of immune responses it produces. If that is promising and we believe that it meets the criteria to proceed to further stages, we go into phase 2 trials where there are a few hundred individuals who it's tested on. And there now you refine the dose that's administered, as well as get more information on the type of immune responses you see from a larger group of people. In phase 2, you generally also see many programs use this as an opportunity to test for a signal of efficacy. So this allows them to say, there's a likelihood of success if we use this vaccine against this disease, but it's not enough information to get a license issued. And once that has been completed, then you go into phase 3 that enrolls typically thousands of individuals, and you're using this to gather all the information that would be required by regulatory authorities to allow for you to use this on a mass scale in public. If you look at the blue column on your screen, this process can take up to 10 years for diseases that are more difficult, like HIV, where we've had to go back to the drawing board several times. It's taken more than 30 years. It's a very well described process that has been set out for this. Several years ago, as we saw the emergence of SARS, the original SARS in 2002, 2003, development of MERS, and the development of the H1N1 pandemic flu vaccine, flu virus that caused the pandemic in 2009, groups had come together to discuss what happens if we have a virus that causes a pandemic and has an unacceptably high morbidity or mortality rate. And these groups had already said, well, we cannot have a development process that takes 10 years to respond to an pandemic. And so discussions went into asking, how can we shorten this process? And so even before the onset of this current pandemic, groups like the Coalition for Epidemic Preparedness and Innovation had already described that means that could be used to shorten the development process. And what you see on the red column on your screen sort of summarizes that, where you have a process of preclinical testing that is much shorter and answers very specific questions about the safety and immunogenicity of the vaccine candidates to allow you to go into humans. The preclinical process can continue even as the phase 1 trials can start, where the phase 1 trials concurrently with some aspects of the preclinical studies. The phase 1 trials themselves would then be used to very quickly assess for safety. And even before the conclusion of that, as long as you have a plausible safety signal that allows for expansion, then the regulatory authorities would allow you to move to phase 2, where more people could be tested, and you could also get a safety signal. And similar to what has been done with the preclinical and studies, the phase 2 trials could then run concurrently with the phase 3 trials in an overlapping fashion to collect as much data as possible very quickly. Overall, what this is trying to say is that the level of data that would be required to license would still be the same. The speed at which you would go into it would be much faster than has previously been done, but the risks are greater for those who are participating as well as for those who are in the trial. But it does give you the opportunity to reach where we are seeing vaccines approved for use in less than a year since the development process began. In addition to what has been done to innovate around the clinical development and the preclinical development, there's also been a push for companies that are capable of producing vaccines to go into trials at risk. And they will then start the manufacturing process even before the data has been produced. And this is usually unacceptable for companies because they do not want to go through a process like that and then at the end of the day, loss. So governments really had to come into play to subsidize these costs of the process and give everyone the opportunity to do the development of the manufacturing capabilities. Didn't have the final data just yet. And so what you see here is that initially we anticipated this process would take 2 years, but by shortening the time period for regulatory reviews, as well as shortening the period for manufacturing and availing the vaccines for distribution, then it's come down to 1 year. So now I'd like to focus specifically on the that's available in Kenya, and this is the ChAdOx1 nCoV-19 vaccine. As I said earlier, this vaccine was developed by the University of Oxford and consists of a non-replicating chimpanzee virus vector, which is called ChAdOx1. This is an artificial virus. Sorry, Kennedy, could you go on mute? Kennedy, if you could go on mute. Right. So the vaccine itself contains the genes that code for the glycoprotein of the SARS-CoV-2 virus, but doesn't actually contain the virus itself. Once the vaccine has been injected into humans, the artificial chimpanzee adenovirus penetrates the cells of the body and uses the cell machinery to produce the spike protein for SARS-CoV-2. These proteins are then presented on the surfaces of the cells that produce the protein. This gives your immune system the opportunity to recognize them as foreign and produce both an antibody response as well as a T cell response against those proteins. What this means is that once an individual is exposed to the actual SARS-CoV-2 virus, then the antibodies and the T cells can work to do two things. The antibodies can bind and neutralize the virus, which is free in, which may be freely circulating in the system. And for the virus that has infected cells and is replicating, then the T cell responses would be capable of finding and using a killer T cell response to destroy the infected cells, thus preventing, on the one hand, infection, and where infection can't be prevented, the prevention of disease, severe disease, can be achieved. So the AstraZeneca team set out to evaluate vaccine in adults, and the trials were carried out in the UK, Brazil, South Africa, and in Kenya. Now, when looking at the trials that were used to produce the data that has been allow for the emergency use authorization, the data comes mainly from the UK, Brazil, and South Africa. So the sites in the UK and Brazil were to produce efficacy data in addition to the safety data. So they were able to demonstrate the prevention of symptomatic infections in both the UK and Brazil that was used to the licensure of this product. The data from South Africa, because of the time when the trial started, was after their first wave. The data was able to provide for safety data, but could not contribute to the efficacy data for this particular vaccine. And when looking at the data from Kenya, the trial in Kenya actually kicked off late last year, and therefore the data from that trial was not available for review by regulatory authorities when the initial authorization was given. The trial in Kenya is still ongoing, and we do hope to see the data coming out later this year. So looking at the specifics, the trials that were conducted in the UK, Brazil, and South Africa, you see that the early phase 1-2 trials in the UK enrolled about 1,000 people. So as I said, in the development process now, they've gone from individual phase 1 trials to trying to accelerate development by allowing these trials to overlap. So the phase 1-2 trial was the first in human trial, and that enrolled about 1,077 people between the age of 18 and 50. The population was healthy adults, and a single dose was initially given for this trial, and the vaccine against SARS-CoV-2 was being compared to a meningococcal vaccine, which is already licensed. During this trial, they made a decision that perhaps one dose would not be enough, and they amended the trial protocol to allow for administration of 2 doses. This allowed for them to go in phase 2 trials, which they combined with the phase 3 trials in the UK, as well as Brazil. And in the phase 2-3 trials, they enrolled over 12,000 people in the UK and over 10,000 people in Brazil. And during these trials, they administered in 3 different age categories. So healthy adults between 18 and 55, older individuals between 56 and 69, and then elderly individuals above 70. So all those were the different age categories that were in. A similar approach was taken in Brazil and in South Africa as well. When looking at the dose regimen, all these were administered in a 2-dose schedule, and it was 1-to-1 randomization, where half the participants received the SARS-CoV-2 vaccine, and the other half received meningococcal vaccine in Brazil and the UK, and in South Africa, they received a placebo. Several happened during the conduct of this trial. As you can imagine, mistakes are much more likely to happen in this accelerated approach. And one of the things they noted was that a significant number of participants in the UK especially were found to have received half a dose as their first dose, followed by the standard dose that had been recommended. This problem did not happen in Brazil or South Africa, where the standard doses were administered. In addition to that, because of the timing, although they were hoping to have a dose interval of about 10 weeks, they ended up with a situation where more than 50% of individuals received the second dose more than 12 weeks after the initial dose, and this was mainly in the UK as well. So looking at the procedures that were carried out to look at to assess for safety and efficacy of this vaccine against SARS-CoV-2, they followed a standard procedure for assessing safety. So there are base assessments where you confirm that an individual is otherwise healthy before they get into the trial. There are blood samples that are routinely drawn at baseline to check for liver, renal function, hematology, and physical exams are generally done alongside a medical history at the baseline to just confirm that an individual is healthy. Following that, individuals are followed up periodically to collect safety information, which is divided into either what solicited events, these are the ones we expect to see. So because this viral vector had been used before for other vaccines, we knew that there's a likelihood of it causing pain at the injection site, change in color on the skin at the injection site, as well as swelling. And systemically, it's been associated with the onset of headaches, fevers, chills, shivering, sometimes muscle aches, as well as joint pains. So rather than wait for participants to report these, the procedure was actually call them in and ask them, did you experience these events? So we are very sensitive procedures for capturing those events. Beyond that, for testing for efficacy, they were looking for 2 things. Number one was symptomatic SARS-CoV-2 infection, so symptomatic COVID-19. And in the UK and Brazil, as well as South Africa, they had different definitions for what symptoms they'd be looking for COVID-19, but these generally included coughing, shortness of breath, loss of smell or taste, as well as in some cases, some abdominal symptoms, other symptoms of upper respiratory tract infections, and then general constitutional symptoms like fatigue or malaise. Once a person was found to have any of these symptoms, then a nasal swab was drawn and tested for whether it could be due to COVID-19. If there were no symptoms reported, there was a process for collection of swabs to try and pick up asymptomatic infections. And these were self-administered in the UK and collected during clinic visits in Brazil and South Africa. When trying to define the outcomes that would be used to measure efficacy, the primary outcome for efficacy against COVID-19 was virally confirmed symptomatic COVID-19, which would be defined as a nasal swab that's positive on nucleic acid amplification test, that's PCR, combined with a fever and those that I've described already. In addition to that, because there was systematic collection of adverse events and the safety outcomes were defined based on those. For the safety points, there was a global analysis of all 4 studies where all the data was pulled into one to look at safety. And a similar process was followed for efficacy, but this was discussed beforehand with the regulators to see what would be acceptable. This slide just highlights the distribution of the participants and their baseline characteristics. And what I would like to highlight here is that they did have a representative sample in terms of age, ethnicity or race, sex, as well as the background comorbidities for individuals who contributed to the efficacy assessments. Looking at the results, the overall efficacy that was picked up when looking at the data from Brazil and the UK was a composite of all the different efficacy endpoints that were tested, and it came to about 70.4%. For those individuals, during the sub-analysis, they found that those individuals who first accidentally received a low dose and then later received a high dose, the efficacy was 90%, so more than was seen for those who received the standard dose. And for those who received 2 standard doses, it was closer to 60%. When looking at the efficacy endpoints, more specifically, you can see that they did find incidence cases for across the different sites in the UK and in Brazil. So 90 cases in the UK, 102 cases in Brazil, and were able to measure this based on distribution between those who received the vaccine and those who received the control product, which for those 2 sites was, for those 2 countries was meningococcal. So when specifically looking at safety, I've already said they pulled the data from the 4 studies. Some of the major safety events that were picked up during the conduct of the trial was a case of transverse myelitis that resulted in a study pause and a requirement for a review by what we call the DSMB, or the Data Safety and Monitoring Board. It's an independent group of individuals who come together to comment on any safety events that have occurred during the course of the trial. They do not work for the sponsor of the trial and are not in any way linked to the conduct of the trial. Their view is given independently. What was concluded after review of the data on the initial case of transverse myelitis that caused the study pause was that it was actually not related to the administration of the vaccine. In South Africa, they did note an individual who had what we call a grade 3 fever of 40 degrees, and this also required review. That was linked to administration of the vaccine, but the participant recovered rapidly following hospitalization. During the course of the trial, there were 2 additional cases of development transverse myelitis. One of those individuals had received the control vaccines, that's the vaccine against meningococcal disease. And this was 68 days after administration of that vaccine. The other individual had received the SARS-CoV-2 vaccine, but was found to have previously unrecognized multiple sclerosis, which was pre-existing and was thus linked to the case of transverse myelitis. Yeah, I think I covered that quite comprehensively. So based on that safety and efficacy data, the European Medicines Agency gave emergency use approval for this vaccine earlier in the year, and the WHO proceeded to review the vaccine and give a positive opinion as well in February. Following the WHO review, our own Pharmacy and Poisons Board reviewed the available data and have been able to give emergency use authorization as well. And that is what triggered the availability of the vaccine in our through the COVAX program. What has been since the start of the programs around the world, I think the most prominent safety signal that has been reported was reported last few weeks, where blood clots were reported in recipients of the vaccine in several countries in Europe, and some countries made the decision to halt their vaccination programs, and these were countries in Europe as well as in Asia, based on those reports. The European Medicine Agency, which is the regulatory authority for countries within the European Union, submitted this, what we refer to as a safety signal, to its body that looks at these cases and makes decisions on or recommendations on the continued use of the vaccine. Their body is referred to as the Pharmacovigilance Risk Assessment Committee. And just yesterday, that committee said that while it's still investigating the ongoing reports, their position is that the benefits of the vaccine actually outweigh the risks that have been reported so far, and thus they recommend the continued use of the vaccine. So looking at what has actually been reported so far as the reports of blood clots from different countries, there have been 37 cases of individuals that have developed either pulmonary embolisms or deep venous thrombosis following administration of the vaccine. And for the time period between administration of the vaccines and development of these conditions, these vary from person to person, with some being pretty soon after administration and others several weeks later. Of these 37 cases, this is against a background of more than 1 million doses that have been administered in the countries where those reports are coming from. And on this, the investigations will be trying to assess, number one, is there a temporal relationship, and what kind of temporal relationship exists between administration of the vaccines and the occurrence of these events? Number two, is a logical basis for this, so starting to look again at the preclinical studies in animals as well as the clinical trial participants themselves, and then looking for any possible pre-existing conditions in the individuals who suffered these events. Lastly, from an epidemiological standpoint, trying to see whether there's an increased number of reports that are coming in following administration of the vaccine than would be expected without the vaccine. So many of these countries keep records on these diseases and will be able to tell whether there's an uptick in the number of cases following the introduction of the vaccination programs. So all this will continually be looked at, and we'll continue to monitor what the regulators are saying. But in any case, at the end of the day, what this will inform is the benefit versus the risk assessment for these vaccines. For a disease that has already killed in excess of 2 million people over the past year, will the number of cases of reactions outweigh the benefits that these vaccines could bring in terms of ending the pandemic? And so those are some of the questions that would typically be asked in those fora. Just yesterday, there was a publication from in the New England Journal of Medicine that looked at data from South Africa. So as I said, the South Africans were continuing with their trial, and one of the things that we were very interested to see is what happens with use of this vaccine in a setting like South Africa, where the predominant strain of SARS-CoV-2 is the B1.351 variant, which is common with the South African variant. And what Shabir Mahdi and his team have reported this week is that for mild and moderate SARS-CoV-2, they were not able to demonstrate any efficacy of this vaccine against that particular variant, which means that we now are very keen to check what happens when they do the analysis for severe disease, which is the endpoint we are most interested in, to see whether, even though it does not prevent mild to moderate disease, does it prevent severe disease? Or is it the same where there is no efficacy for severe disease? So these data were just reported this week in the New England Journal, and we're waiting to see their follow-up reports, what they've seen for severe disease. So I'd just like to conclude by saying that, number one, as far as the case for use of the vaccines to address the pandemic, I think there was a strong case based on the bad disease and the number of severe cases of diseases and death that were occurring around the world, in all countries, including in Kenya, where we've lost more than 2,000 people in less than a year to this disease. The second point is that the data that had been collected from more than 25,000 individuals during the clinical development did support the introduction of this vaccine, and that's the safety data alongside the efficacy data. And then lastly, when it comes to the data that has been seen after the approval, when it comes to these safety events, as well as the efficacy data that's coming out of South Africa, I think the data we have so far is data that will need to continually be looked at. We'll continue to assess the risk versus benefit of use of this particular vaccine. But at the moment, the data does support the decision to continue with the vaccination program and expand it further across the country. So I'd like to stop there and hand it back to Dr. Kabata. Thank you for your attention.
Thank you very much, Dr. So there are questions flowing in fast and furious. So maybe we can look at, so there's one on elaborate on the viral vectored vaccine. What do you mean by that? I think we can start with that. And then what happens if you miss your second jab? Then, yeah, I think let's do the first three. So one, you have an immune response based on antibodies and T cells. So how long does that last based on actual COVID exposure versus the vaccine? And so I guess the question is, if you have gotten COVID and lived to tell the story, so do you need a vaccine? Two, if you need a vaccine and you miss the second dose, what does that mean? And three, since this is a vectored vaccine, what does that mean? And then you can do another three.
Okay. So thank you for the questions, Dr. I'll start with what is the recommendation if you've already suffered from COVID-19. So we've seen several individuals who've been infected and recovered and produced what we call neutralizing antibody responses. And this means that the antibodies they produce are actually capable of neutralizing the virus when tested in the lab. And so this was seen as a very promising avenue, which some have also tried to use to develop new products that can be used for treatment. When asking the question, protect them in the future, it is very difficult to say, because what was noted with the antibody responses following natural infection is that they waned quite quickly over a 3 to 6 month period. And for many individuals who had recovered, during that time, you then had them walking around without any antibodies in their system that were detectable. The question was, if they are exposed, do they have a memory immune response that would allow them to quickly produce those antibodies to protect? The answer is we don't know. And that is something that's still being studied. For several individuals, aside from those who produce these very effective immune responses, there were others who were seen to produce antibodies that were not likely to be protective because they were either binding antibodies or were of a low titer and or lasted for a very short period of time. And so those with poor quality immune responses, these mainly being elderly or individuals with pre-existing immunosuppressed states, then for those ones, it's unlikely that we'll see effective protection in the future. I think the most promising aspect from public health data is that we have not seen a massive number of people who've been reinfected and suffered severe disease. So that gives us some hope that existing immune responses that they develop after infection could be protected, but we just don't know for how long, if that is the case. When talking about, well, do those people then need to be vaccinated? The current recommendation is that individuals who suffered from COVID-19 and recovered should be vaccinated. And this is more or less like an insurance policy to say, because we don't know whether you're protected or not, you should receive a vaccine to be assured of protection. To your other question on what happens if you miss the second jab. So right now in Kenya, the recommendation from the government is that the 2 doses should be given 8 weeks apart. And if you do not manage to get your second jab on time, the 8 weeks, then you should get it as soon as possible after that. What was seen in the trials that I've highlighted for the AstraZeneca vaccine is that individuals with a time point of 12 week administration of the first jab tended to have actually stronger immune responses than those with shorter intervals. So I would say you should just go ahead and receive the second dose as soon as possible after the 8 weeks has passed. And then to the third question on what do we mean by a viral vectored vaccine? What we've traditionally done with vaccines is, if you think back to the measles vaccine, which we've had now in use in our country, for example, for nearly 40 years, we used to have live vaccines that had been weakened. And so you just take the pathogen itself, weaken it in the lab, and give it as a vaccine. And diseases like measles that tended to produce a very strong immune response. So that's what we use for measles and for polio and several other disease areas. Over the years, we started using killed pathogens. So when you think of pertussis that causes whooping cough, we use a killed whole cell bacterium because it would be very dangerous to give a weakened pathogen in that case. And we still do protection with those killed pathogens. Unfortunately, these live and killed pathogens, when we give the whole pathogen, they tended to have a very poor safety profile. And so the push was to develop what we call subunit vaccines, where you just give a small part of the pathogen, and therefore it's easier to produce, it has a better safety profile, and it can be used much more quickly. One of the strategies for using these subunits is to put it in another virus, to put the part of virus, like the part of SARS-CoV-2 that you're interested in getting an immune response again, into another virus that causes no disease in humans. So a chimpanzee adenovirus was modified by the University of Oxford so that it replicates itself. It's alive, but it doesn't replicate, but it can get into the human cells and use the cell machinery to produce proteins. And you can then engineer it to produce the proteins for SARS-CoV-2. And that's what's happened. So a gene segment from the SARS-CoV-2 was put into this chimp adenovirus, which causes no disease in humans. And that adenovirus is used as a vehicle to then get into the human being and use the human's own to produce the protein we are interested in, which is the surface protein for the SARS-CoV-2. Does that answer the question?
Yeah, I think Kioi just mentioned, so how long does the immunity then last for after the vaccine? Is that known?
So what we do know is that for at least 3 months after vaccination, the immune response is still as strong as we would hope to use for protection. Unfortunately, we have limited data. The way we usually find out how long an immune response lasts is by following vaccinated individuals longitudinally, so over a period of time. For new vaccines, it's impossible to say how long it will last. But if you look at what we've had with vaccines, so for example, for the hepatitis B vaccine, which was developed in the early 80s, initially the thinking was, okay, we know it can last for a year. Then you follow people up for 5 years and say, oh, the immune response is still strong. We think it can last 5 years. And then after 10 years, the same thing is reviewed, see what the immune responses look like, see whether people have immune memory to respond by producing more antibodies when they encounter the virus. And you see, oh, this is continuing to be there. And just a few years ago, a committee of experts met and said, look, for the hepatitis B, it's more than 30 years. It looks like it's a lifetime of immunity once you're vaccinated. And it's the same thing when you saw the change in the recommendation for yellow fever from 10 to a single dose lasts a lifetime. So at this stage, we can't say. What we will need to do is follow people up to find out just how long protection lasts following use of the vaccine.
Right, thank you very much. I know our time is up, but I'll ask people's indulgence because I think there's another 3 which I want to ask you. So one, what happens if you get the vaccine and you have COVID, either symptomatic or asymptomatic?
Mm-hmm.
And then 2, regarding blood clots, if you already have had DVT, pulmonary embolism, you have a history of that, is that a good idea to have? And 3, should you take, ASCAD prophylaxis against, Jaffa is asking, should we then all just take a small prophylaxis against blood thinners? Any comments?
So to your first question, what happens if a person gets the vaccine when they're concurrently already infected with the SARS-CoV-2 virus? Now, the recommendation is that anyone who is symptomatic should not receive the vaccine because the vaccine wouldn't add any additional protection. And so if SARS-CoV-2, the vaccine will not benefit you once it's administered. What that means is that if a person is known to be infected, they shouldn't get the vaccine at that time. It is recommended that they get the vaccine after they've recovered and a period of somewhere between 1 and 3 months has passed. For those who are asymptomatic and they don't know, well, in that case, then they are basically walking around like the rest of us, and they should be vaccinated. But an individual is suspected to have SARS-CoV-2 based on symptoms, they should seek medical care rather than receive the vaccine, because if they are infected, it would not benefit them at that stage. Your second question was around your other question was around prophylaxis for clots.
Yeah, and also people who already have clot issues.
Right. So for individuals who have clots, the current recommendation is that these individuals should be receiving care. So if a person is on blood thinners or is already on follow-up and management by a physician, then if they're in a controlled state, then they can receive the vaccine like everyone else, because their risk of COVID is still present. I would think that the recommendation will go forward to include close follow-up of those individuals to ensure that there's nothing untoward that happens. Know more about what's happening with these reports from Europe. But the current recommendation does not prohibit administration of those individuals, especially if they have other comorbidities that make them susceptible to severe COVID-19. When looking at the question of should you take blood thinners ahead of receiving the vaccine, I think I would strongly recommend against that. Number one, you're opening yourself up to receive a second product that also has adverse events associated with it. So you're increasing your risk of a negative outcome because you're taking something that you might not need. In the case where you're asking, would that prevent, if, for example, the vaccines are associated with this and it's a true association, would that prevent clots from developing in this individual? There's no evidence of this. So the prophylaxis, I would strongly recommend against prophylactic antiplatelet agents. And just to add, when looking at the initial reports of those 37 cases, there were some individuals who were reported to have blood clots, but at the same time had low platelet counts at the time when the reports were made. So it's unlikely that there's any evidence to support use of antiplatelet agents before vaccination.
Thank you very much. Last but not least, I think there's a question of, does risk matter? I think your data showed risk doesn't matter. And then there's a question of how do you explain the inverse? The guys who got half dose by mistake, then got 90% efficacy. How do you explain that? And lastly, any comment on the Johnson & Johnson one, which seems to be more efficacious?
So starting with the question of, is there a possible explanation for inverse finding of higher efficacy in those who received a first dose that was half of what was recommended? There is no definite answer to that at this stage. So there's still questions around whether the data in animals that could support that, or any data in humans that could point towards why that is so. What I can say is that we have seen similar outcomes in other disease areas. So I worked for some time on malaria vaccine development, and we saw for one of the malaria vaccines candidates that when we did have a similar situation with fractional dosing, and ironically enough, it happened in that situation as well, because they sort of ran out of doses and decided to split the doses for the trial. The responses were much higher for individuals who receive half dose than a full. And the possible explanation that has come out when the analysis has been done from those individuals is that the fractional dose or the half dose tends to select for the much more potent B cells with germline. And therefore, when these B cells are boosted by a full dose later, they tend to produce a much stronger and directed immune response with antibodies that have stronger affinity maturation. And therefore, it's a better. And so this is very theoretical, even in that space for malaria, but I think for the malaria vaccine, they are currently testing that proactively in a well-designed clinical trial to see if it actually pans out, whether the fractional dosing can work. I would expect that the team would take this forward as well. Could you remind me of that question?
The third question is the Johnson & Johnson.
Right. I think the Johnson & Johnson has several promising attributes that would be beneficial to like ours. Number one, it has the ability to develop the immune response that's been noted following one dose rather than two. So that's very beneficial for a vaccination program. It is relatively affordable compared to the other vaccines being produced in the US. There's sites producing that vaccine in South Africa, which means that when looking at regional availability, it might be beneficial to us. In addition to that, we have seen that when the teams in South Africa have tested the Johnson & Johnson vaccine against mild and moderate disease from the South African variant, it seems to be protected. And so there are several data points that we are looking at. Well, that looks like it might be beneficial, but as it is not available to us at this time, it's not something that we can discuss in comparison to what we currently have available.
Well, Dr. Ali, thank you very much. I feel like I've just upgraded 1 year worth of knowledge in a few minutes. And I'm sure just seeing the comments, I think this has been just so tremendously helpful. Mushai, thank you for the link. This is great. Thank you very much. Frank, I'll hand over to you for any final comments.
Thank you for having me.
Thank you very much, Dr. Kioi. Again, thank you very much, Dr. Kibata. Now I want us to welcome our president, Dr. Korir, for the announcements. Then I'll close the sessions after that.
Thank you very much, Frank. Thank you, Dr. Kibata and Dr. Kioi. Thank you. Good to see you, I think Dr. Kioi was my classmate at one point. So thank you very much for that. And, very enlightening, as Kibata has said. Just quickly, just some few announcements as OSK. I'd just like to congratulate Dr. Gishuhi for being, taking over as the chair of the department in the University of Nairobi. So congratulations from OSK. Thank you, Professor Karimori, for the good job, well done, and handing over the mandate to Dr. Gishuhi. Again, just to remind everybody, to pay up your subs. You saw when we were starting, we're just reminding everyone, 31st of March shall be our deadline. So please pay up. We will only give CPD points for those people who've paid up. Thank you for all those who have paid already. We've had, the last time we checked, 38 people have paid up subs. So that's probably the highest number paid up by this time. So I'd like to encourage, if you haven't, please do so. Finally, I'd like to bring to your attention that now we have active committees at OSK. I'll briefly run through them so that you know what exactly they do and who the chairs are. So the first one is Professional Standards and Ethics Committee. This is basically a committee that will look at ethical issues around ophthalmology, whether there are things that we need to be doing better. I saw a question today by Dr. Kibata saying, should we have biometry from anyone, all the patients doing cataract? Should we transition to CCC? What are the standards around that? This is a committee that looks at such things, and also to look at any ethical issues that relate to a particular individual. And so we have seasoned team that looks at that, headed by Dr. Gishuhi. The second committee is the scientific committee. I think scientific committee has been there, and their main mandate is to basically populate at the end of the year when we're having our annual conference. They're in charge of the scientific aspect of all the papers, reviewing and all that. And they've also been mandated to try and enhance research output amongst OSK members. So they're looking into meeting and discussing ways in which that can be improved. This is headed by Dr. Mutie. The other committee is Education Committee, and this is a committee that is in charge of all the educational material, particularly throughout the year. And as you can see, Dr. Kalu, Dr. Nyakagwa, that's the main team that's in charge of that. Dr. Kalu is the chair, and they're doing a wonderful job. So they'll be mainly running the CMEs. The other committee is the Annual Meetings and Communications Committee. This is basically the committee that deals with how the conference is going to run. There's so much, where is it going to be? How is it going to be arranged? What are the colors? All this kind of thinking, putting up the conferences together. So this is a committee that shall be headed by Dr. Mushai at the moment. And they're also in charge of websites, any forms of communication. So that's headed by Dr. Mushai. We have Business and Advocacy Committee, which is involved with liaising with insurances, Kenya Health Federation, anything that relates to ophthalmology and business. Those will be the team that will be doing that, as well as advocacy and bringing eye care to the thoughts of members of parliament and any decision makers, making sure also that we're in all stakeholder forums. So this committee is headed by Dr. Nyenze. And finally, we have international relations and member support. This is headed by Dr. Munira. Their main mandate is basically to make sure that all the members, you're supported as a member. We have so many things happening in our lives that we might need support of each other. And this is the committee that makes sure that happens. We're sensitive to everybody's needs, and so that we can get together and help support each other as ophthalmologists. They will also be mandated in making sure that we are well represented in many forums, such as the AAO, American Academy of Ophthalmology, ICO, COEXA. Those are our links, and Royal Society of Ophthalmology. All these international bodies, they're basically the ones in charge of that. So in a nutshell, those are the 6 committees. We shall be publishing them for your reference. Thank you once again for participating in the CME. And I'd like to hand over back to Dr. Frank. Thank you.
Thank you very much. Thank you very much for Korir and the presenters, Dr. Kibata and Dr. Kioi. The only thing I want to remind you that we'll have a CME again on Thursday. The coming week, the flyers will be out by early next week. It will be about glaucoma. Dr. Nyamai will be presenting. It will be about the advances in glaucoma surgery. So thank you. Have a good evening. Hope to see you again Thursday next week. May God bless you. Thank you very much.